As Stephan said, my name's Patrick Turley. I'm a postdoc in Ben Neal's lab at the Broad Institute. And I'm going to be talking about this method that we developed. On the screen right now, I'm on slide 2, is a list of all the people who've been involved. And so I think some of them are even on this call. And so it's been a big effort by a bunch of people. This is already posted by our archive. And it's forthcoming in Nature Genetics. So the motivation for this project is that in order to successfully do GWAS, you need very large sample sizes. And if you want to make predictors based on GWAS results, you need even larger sample sizes. And in a lot of cases, these sample sizes just aren't available for traits that we're really interested in. In a lot of cases, as well, though, we will have summary statistics for traits that are genetically correlated with each other. Like as you can see in the picture that I'm sure you've all seen from Bullock-Sullivan's LD score paper, we actually see that a large number of traits are very strongly related. And so the goal of this project was to boost the power of our GWAS results by incorporating information from multiple genetically correlated traits. And there were 3 obstacles that we wanted to overcome when we designed this. First, we wanted a method that didn't require individual level data, so it would operate just on the summary statistics. We wanted it to be a method that was robust to overlap, potential overlap in the summary statistic samples. And we wanted it to be something that was computationally feasible, even for a large number of traits. And so here's the underlying model, very briefly, that you should have in mind when we're thinking about MTAG. So first, imagine that we have T traits. And so we have a vector beta, which is the vector of marginal effects of SNP J or of the outcome on SNP J. And so this is different than what you might see. In a lot of cases, we'll see a vector beta, and what we mean is that it's one trait. It's the effect of all of the SNPs in the genome on a single trait. In this case, when I say beta sub J, I mean it's the effect of a single SNP, SNP J, on all T traits. And so this is a T by 1 vector. When we do GWAS, we estimate, we don't know what the true beta is, and so we have to estimate it. And so we have some observations, beta hat, which is the truth plus some noise. And assuming that we're using something like OLS or an unbiased estimator of the effect sizes, then this estimation error from the GWAS should have mean 0 and a variance-covariance matrix, which I'm going to define to be sigma. And so if the samples for all the different traits that we have are non-overlapping, then sigma will be a diagonal matrix. But if we have overlapping samples across these different sets of summary statistics, then the off-diagonal elements of sigma are going to be non-zero. And so we're going to be really flexible with our sigma matrix here, and we're going to allow the off-diagonal elements to be non-zero. And this is going to be the element of the model that will allow us to have a potential sample overlap. And so MTAG is a random effects model, and so we're going to assume, so I said this is the format of the error term of our beta hats, and we're going to also assume that the true effect sizes are drawn from some distribution. And we're not going to make really strong assumptions about the distributional form of beta, but we're going to make 2 moment assumptions. So the first one is that in expectation, the betas have mean 0. I think this is a justifiable assumption if we think of the reference allele that we're choosing is chosen at random. And so, so the sign of beta is going to be randomly assigned, and so the mean is 0. And then the strong assumption of this model, and the workhorse of the model, is that we assume that the variance of beta is equal to this kappa omega. And so notice that there is no J subscript on omega. What this means is that the relationship between the effect sizes throughout the genome is homogeneous for all SNPs. And so this is actually quite a strong assumption. An example of a setting that would violate this assumption is, let's say that you're considering 2 traits, And let's say that for most SNPs, the SNP is associated with both outcomes, but there's some mass of SNPs as well in the genome that affect the first trait and not the second one. For those SNPs, the relationship, they're going to be uncorrelated. If the association of the SNP with the first trait's exactly 0 and the other one there's some distribution to it, then they're going to have a 0 correlation. And so as a result, the omega for those SNPs can't be the same as the ones where they're both associated. And so that's going to be a problem for us. I'm going to spend more time later in the presentation talking about exactly what the consequence of that problem is, and how big a problem it is, and how we can avoid it. But this is the big assumption of MTAG. We usually refer to it as the homogeneous omega assumption. And so this is an important thing to keep in mind. Moving on to slide 7. So because we're just relying on the first and second moments of the beta, MTAG is a moment-based method. And so we're going to, you can look into the paper if you want the details of the derivation. But based on those 2 moments, you can derive an estimator of for the effect of SNP-J on any individual trait T. And after a little bit of work, you end up with this formula that is on this screen. At first glance, it looks quite complicated, so I'm going to try to break it down a little bit, break down the intuition. And so first notice, the denominator there is just a quadratic form, and so it's just a scalar, and so we can mostly ignore it and just think of it as a normalization factor. Let me describe all these terms to you first before I break down the pieces of this equation. And so first, beta hat MTAG, that's our estimator for SNP-J on a single trait T. The bold omega sub T, that's the teeth column of the omega matrix. And so if you recall, omega is the variance covariance matrix of the betas. And it turns out that this is going to have the same interpretation, or approximately the same interpretation, as just genetic covariance. And so cap omega sub T, since it's the teeth column of omega, is just the vector of genetic covariances between trait T and all other traits in the model. Omega sub TT is just the teeth diagonal element of omega, so it's the variance of beta sub T. And then omega I've already described, it's the variance covariance matrix of beta, and this cap sigma, if you recall, is the variance covariance matrix of the error term of the GWAS estimates. And so breaking down this equation, so on slide 8, you can see on the right-hand side, we have the beta hats, which are the GWAS estimates. And on the very left, we have this vector of the genetic covariance terms. And so, one other thing to highlight before I explain this. If you look at this, you can see that this thing is just a weighted sum of the beta hats. And so this is going to feel a lot like meta-analysis. In fact, this reduces to just simple meta-analysis in the case that there's no sample overlap and all of your traits are perfectly correlated genetically. And so we just have to think about what are the weights that are being assigned to each of the summary statistics for each of the traits. And so looking at this first term here, omega sub T, this is the genetic covariance. And so what this means is that if the genetic covariance between a pair of 2 traits is high, or between T and some other trait is high, then you're going to assign more weight to that trait. And so that's what we'd expect, because if there's a high genetic covariance, then there's going to be more information about trait T in the other traits, and so we maybe want to give that more information. And then this middle term is a term that's, the term that's in the inverse is a term that's capturing how much noise there is in the residual of the MTAG estimators. And so this, you can think of this in 2 pieces. So first, let's look at the sigma piece, because that's the easiest to understand. That represents how much variance there is due to estimation error. And because we're taking the inverse, we can think of, if there's a big term in the middle, that's going to downweight how much information we're carrying from a particular trait. And so if there's a lot of noise for in the GWAS estimates for a particular trait, That means sigma will be large, and that's going to put less weight on that particular trait, just as we want, because we don't have a lot of confidence in those estimates. We don't want to put a lot of weight on it. The first term is the residual variance in the genetic effects. And so let's say that you have a pair of traits that are genetically, have a very low genetic correlation, you know, like maybe 0.1, 0.2. Well, there's perhaps some information in those effect sizes about the effect on trait T, but there's also just a lot of uninformative noise. And so this term here is capturing how much noise there is. And so if two traits have a low genetic correlation, this is going to downweight the effect of, or the weight assigned to that secondary trait. And so I think that each of these pieces are pretty intuitive. It puts more weight on things where there's a high genetic correlation and removes and puts less weight on things where they don't supply a lot of information about trait T. And so there's some good news and there's some bad news about MTAG. So let's start with some of the good news. First off, MTAG will correct for potential sample overlap. And it does this using LD score regression. So it turns out that the elements of this sigma matrix here can be estimated using the intercepts from LD score regression. And I'm not going to go into detail about why that is right now, since time is limited. But the details are in the paper, and we can maybe chat in the discussion period afterwards if people have specific questions about that. So MTAG can correct for sample overlap, even if the overlap is unknown. Another really powerful property of MTAG is that MTAG will always decrease the mean squared error of the estimates relative to the GWAS estimates. This holds even if the model is misspecified. And so as I pointed out in the beginning of the presentation, there's this strong assumption of the homogeneous omega, but we can prove that even if the homogeneous omega assumption is violated, then the average mean squared error across the genome is going to decrease. What this will also mean in general is that if you create a predictor based on MTAG results, that predictor will in general have more predictive power for the trait of interest than a predictor based on the GWAS summary statistics that were used in the estimator. And so, so MTAG is going to be really great if you want to create a predictor. Another nice property of MTAG is that under the assumptions of the model, so if the homogeneous omega assumption holds, then the betas that MTAG produces have a trait-specific interpretation. And so if you're passing in a GWAS summary statistics for depressive symptoms or something like that, then MTAG should spit out betas that also correspond to depressive symptoms. Again, though, this is going to rely strongly on the MTAG assumptions holding, and so so you know, to the degree that you think that the assumption holds, this is going to be true. This figure on the right is just highlighting this decreasing mean squared error thing, and so the black line at the top is the mean squared error under a GWAS, and then under different settings. So if there's different genetic correlations between a pair of traits, you pass into MTAG, or if there's different levels of power of the GWAS summary statistics that you pass in, you can see that it's always less than the baseline, the mean squared error of the GWAS. So here's the bad news. If the model is misspecified, you can actually get a substantial bias in your estimates. And that bias can lead to inflated type 1 error rate and inflated false discovery rate. It means that instead of having this joint interpretation that I was highlighting on the previous slide, I'm on slide 11 for people who are following along with their own slides. But yeah, so this joint, the trait-specific interpretation that I was describing on the previous slide no longer holds, and our effects and the betas that we have here and the p-values correspond to a joint test of, you know, a SNP the SNP being associated with at least one of the traits that you've included, although you don't know which one, and so maybe it's of limited interest and limited use. So the figure on the right, what I've done just to highlight how bad things can get is I've considered 3 different scenarios. And so what I do is I fix, so this is for a 2-trait model. And so you can see on the x-axis, I have the genetic correlation between the 2 traits that are included. And on the y-axis, I have a statistic that I've called the max FDR. And what the max FDR is we consider a big range of settings, and specifically what I call a multivariate spike and slab setting. And so there's some probability for each SNP, there's some probability that it's null for both traits. There's some probability that it's associated with both traits. And there's some probability that it's null for one and non-null for the other. And so I search over the space of all genetic architectures that have this fixed genetic correlation, and I say, how bad can I get the false discovery rate to be in this class of distributions? And so you can see that, like in this green line here, that corresponds to a case where you're low-powered for the trait that you're interested in. You have a mean chi-squared of 1.1. Just to kind of give you a baseline of what that means, you'd get a mean chi-squared of 1.1 if you did a GWAS of like depressive symptoms in a sample of 80 or 90,000 people. So it's a pretty small, small GWAS, not doing very well. But then let's say you meta-analyzed it with a really well-powered GWAS. And so, so something with a mean chi-squared of 2. Meaning that it's, you know, a GWAS that's 10 times the size of the depression GWAS. Then you can see that there's enormous potential in inflation. 20% of your discoveries in some circumstances are actually just going to be false discoveries that are being driven by the subset of SNPs that are causal for the second trait, but not for the first trait. And so, so this is a big problem, something that you should be really worried about if you're considering applying MTAG. Another thing you should be worried about if you're applying MTAG is that even if the model is well specified and you meet this homogeneous omega assumption, the errors in your estimate are going to be correlated with the effect sizes of the secondary traits. And so normally when we think of the beta hats, you know, we think, oh, it's truth plus some noise, but we think of that noise as being independent of everything. And so, you know, we can just treat that as you know, as just sort of an independent variable, and it doesn't cause too many headaches. In this case, however, if you can recall from the formula for MTAG, I'm going to go back to slide 9 just so that we can look at that. There are 2 sources of noise in the residual, and the first is the sampling variance, which is what we're used to and tends to be independent of everything that we're concerned with. But there's also the other source of noise, which is just the residual variation of the genetic effects. And so that noise is still present in our MTAG estimates. And as a result, if you apply MTAG and then you want to predict traits that, other than its own trait, so let's say that we've done MTAG and we have a predictor of schizophrenia, and then we want to see if the polygenic score for schizophrenia predicts other traits, we might sometimes get false signals because the residual, let's say that we jointly analyzed it with, I probably should have chosen an outcome that I know more about than psychiatric outcomes. But like, let's say we did a MTAG on cognitive performance, and we jointly analyzed it with educational attainment. Well, educational attainment may be largely cognitive performance, but maybe there are genetic markers that are associated with grit or you know, some other motivational personality characteristics. So if we took our cognitive performance score, it might be correlated with things that are associated with grit, but not actually with cognitive performance. And the only reason that we see an association is because the error from our polygenic score for cognitive performance is correlated with the effect sizes for educational attainment, which then takes us to grit, which then may connect it to other traits as well. The same thing may apply if we're doing biological annotation. And so because the error is associated with, real genotypes that may have their own biology associated with them, if we are not careful, then the biological annotation findings that we get passing MTAG results into something like DEPICT or MAGMA or FGWAS or whatever your favorite biological annotation software is, may just be picking up signal that lives in the residual, and so it might not be actually associated with the true betas that you're interested in. So this is also a big problem. What is especially concerning about this problem is that we don't really have an easy way of testing the homogeneous omega assumption. We can't know if the model is violated or if it isn't. And so even though we have all of these wonderful strong results that I showed you on slide 10, when the model's true, we have all of these concerning results when the model isn't true, and there's not an easy way to tell when it's true or when it isn't. And so moving to slide 12, here are the things that, here are the recommendations that we have for people who are interested in applying MTAG. And so the number one thing that I recommend is always replication. And so if you apply MTAG and you get a lot more hits for the trait that you're interested in, it's always good to be able to have some replication sample to validate anything that you find. You can validate the SNP level results. You can validate the set of SNPs that you find jointly if you'd like. You can even validate the individual biological results possibly, depending on exactly what you're trying to do. And so I'd always strongly recommend trying to replicate anything you can when applying MTAG. If replication is not possible, or even if it is possible, I'd also recommend that you only apply MTAG in settings where there's a low risk of a high false discovery rate. And so if you can recall from this figure, that I showed you some settings where the false discovery rate can get very high. But there are settings where the false discovery rate is more limited. And so, for example, when the genetic correlation between the two traits is high, the false discovery rate will tend to be much lower. And so the rationale for this is, as you recall, the SNPs that really kill us in MTAG are the ones where they're null for whatever trait that we're interested in, but they're associated with the other traits that we've included. So imagine that we have a perfect genetic correlation, a genetic correlation of exactly 1. Then that means the betas are going to be proportional to each other throughout the genome. And what that means is that there can't be any SNPs of the type that I just told you about. And so if the genetic correlation is 1, it means that there are no SNPs that are causal for one and not causal for the other. And so that's good. And so as long as you're within a window of 1, the degree to which these problematic SNPs can exist is limited. Just based on some simulation work that we've done, it seems to be that you're relatively safe as long as your genetic correlation is greater than 0.7. So that's generally a good starting point to think about if your application is a good test case for MTAG. Another important thing is MTAG will the violations of omega will be more, or not the violations will be dramatic, but the consequences of the violations of the assumptions of MTAG will be more dramatic when the GWAS for the trait that you're interested in is low powered, and then you're jointly analyzing it with other traits that are highly powered. And so first thing that you could do is only study traits where the GWAS summary statistics has a very high mean chi-squared statistic. And so it appears that when your mean chi-squared statistic is greater than 1.7 or so, that even if the assumptions of MTAG are violated, they don't lead to a strong amount of bias in your estimates. And so that's one thing you can do, is just make sure that you're moderately well powered for the trait that you're interested in. Another thing you can do is just make sure that the secondary traits that you're including in your MTAG analysis are not more strongly powered. And so if your GWAS summary statistics have a mean chi-square statistic of less than 1.7, then you should make sure that the GWAS summary statistics for the other traits at least don't have a greater mean chi-squared statistic than the trait that, than your primary trait. And so that's illustrated a little bit on the figure on the right. So again, this is the same format as the figure previously. So in the bottom, the x-axis is the genetic correlation between the two traits, and you can see that that's largely decreasing in R beta, in the genetic correlation between the two traits. And you can see in the case of the red line, that is a case where you have two equally powered GWAS. They're both very low powered, but at least they're equally powered. And you can see that the false discovery rate in that case is moderate, but at least it's decreasing in the genetic correlation. I'm just to highlight this point on the far left corresponds to a genetic correlation of 0 between the two. And that's actually the false discovery rate of the GWAS itself. And so if you tried to do a GWAS and only had a mean chi-square statistic of 1.1, your false discovery rate is already at 5% in this case. And so MTAG only decreases it no matter what the genetic architecture is, as long as you don't have another trait that's more highly, is more strongly powered than the other. You can see that immediately when they're both at 1.4, or if they're both at 2, the false discovery rate really just can't get very high. It's less than 1%, no matter what genetic architecture you have. And so, yeah, so those are some just kind of first stage guidelines. I would recommend, in addition to just using these guidelines I gave here, to perform your own max FDR calculations. And so, so you can look at your particular setting, the genetic correlation between your specific set of traits and the power of your specific GWASs, and calculate how bad could the false discovery rate get. That is, I think, currently not implemented in the release of the software that we have online, but it is coded up and we're just testing it right now. And so, so it should be very soon that running these max FDR calculations, you could just do it with a click of the button with the existing software. I wanted to highlight one quick point about, why am I focusing on the false discovery rate? A lot of times when we look at these methods, we spend more time thinking about, what's the type 1 error rate or the family-wise error rate. And especially because MTAG, it's possible to write out settings and genetic architectures where the false discovery rate is quite low, but the type 1 error rate is very high. And so I've been focusing on the false discovery rate because in my head, the question that we're interested in is, we run MTAG and we get a set of genome-wide significant hits. And the question that I worry about, that I have in the back of my head is, what's the probability that if we just took one of these hits that we've discovered, what's the probability that that one's actually truly associated, as opposed to actually being null and just being an artifact of a violation of this homogeneous omega assumption? And that's exactly the question that's being asked by the false discovery rate, which is, of the set of discoveries, how many of them are actually false? So that's my focus there, but I do understand that this is a bit of a philosophical debate as well, so I don't want to spend too much time on it, but just to explain the rationale for why I've been focusing on it in this case. And so in my last few minutes that I have, I want to talk about the application that we have in the MTAG paper. So in 2016, there was a paper that analyzed subjective well-being, depressive symptoms, and neuroticism, big GWAS of all 3 of those traits. And so we've taken that data set, we've added A few additional samples, specifically data from 23andMe is the largest increase in sample size. And so we ended up with a depressive symptoms sample with an effective sample size of about 350,000 people, neuroticism with 170,000, and subjective well-being of 388,000. And so these are pretty large GWAS samples to begin with. And we're going to apply MTAG to the three of these traits together. And we're going to compare the GWAS results to the MTAG results, looking at the number of significant loci, the effective sample size, and the predictive accuracy. So on to slide 19. So slide 19 shows Manhattan plots for the GWAS results for depressive symptoms and the MTAG results for depressive symptoms. And so we can see that we go from 32 hits in the GWAS sample size to 74 after applying MTAG. Based on the mean chi-squared statistic, this is roughly equivalent to an increase in sample size of about 27%. On the next slide, we show the Manhattan plots for neuroticism, and we go from 9 hits in the GWAS to 66 in the MTAG analysis. And this corresponds to about a 55% increase in the effective sample size. And finally, for subjective well-being, we go from 13 hits to 49 after we apply MTAG, and again, a roughly 55% increase in the effective sample size. And so we're seeing pretty large gains if we just look at these summary statistics. If we want to look at prediction accuracy, I said that one of the great highlights for MTAG is that you can see big boosts in prediction accuracy. So moving on to slide 23, what I've plotted here is for each of the traits, I plot the incremental R squared for an LD-PRED polygenic score based on the GWAS summary statistics and based on the MTAG summary statistics for each of the traits. And so by incremental R squared, what I mean is that we take our replication data, in this case the health and retirement study and ad health, which are samples that we didn't include in the main analysis, and we regress the outcome onto just the set of controls. So in this case, principal components and sex, age, I think that's it. And we look at the R squared of that regression. And then we run a regression where we regress the outcome on all of those controls and also the polygenic score. And we look at how much the R squared increases after including the score. And so that's what the y-axis represents. And we see that in all three of these cases, the predictive power of the polygenic score increases. If we want to look at whether these increases are statistically significant, you can look at slide 24. And you can see that these increases in predictive power as we go from GWAS to MTAG are statistically significant. And in fact, these red dots that we have in this figure correspond to the predictive improvement that we'd expect to see given what the increase in mean chi-squared is in the GWAS, in the Manhattan plots that I showed you. And so the mean chi-squared statistics. So if the boost in power that I showed you in the Manhattan plots was just purely an artifact, just noise, then we wouldn't expect to see any increase in the predictive power. And if a big portion of it was just noise, and so it wasn't entirely, so maybe some of it was added information, but some of it was just inflation of the chi-squared statistics, we'd expect to see that the improvement in the polygenic score would just not be as large as expected. But we see that for the most part here, our polygenic scores are improving in power by how much we'd expect them to. And so that's kind of further evidence that that the increase in signal that I showed you is a true increase in signal. And finally, on slide 26, this is showing that the polygenic score for the own trait polygenic score is most predictive of its own trait. And so you can see in the first set of 3 bars, what we've done is we tried to predict depression with the score created for all 3 of the different traits. And so you can see the blue bar is the incremental R squared for the depression score. And we can see that the depression score predicts depression better than the score that's constructed for neuroticism and subjective well-being. We see the same pattern for neuroticism. The neuroticism score predicts neuroticism better than the scores for depression and subjective well-being. And the subjective well-being score predicts subjective well-being better than the score for depression and neuroticism. And so this is also evidence that the claim that we made about how these betas that we're producing have this trait-specific interpretation. And so in conclusion, it looks like MTAG seems to work well in this setting. I want to highlight as well that, you know, we chose this setting specifically because we thought that it satisfied, oh, I must have removed that slide. These three traits that we're considering here are highly genetically correlated, and the GWAS summary statistics for each of them have a comparably mean chi-squared. And so we chose it specifically because we thought it was a setting where MTAG would work well, and it appears to work well. Whether or not it works well in every setting, I mean, it definitely doesn't work well in every setting, but if you choose your settings carefully and do these max FDR calculations and replicate results, we think that the potential for MTAG to really boost power in GWAS and prediction are substantial. There's code available online, as I said. You know, currently the code should work fine for just doing these main analyses, and within the next week or two, it should have this additional functionality of being able to also do the max FDR calculations, and so that's something to also look forward to. So thank you.
Thank you, Patrick. Thanks so much. Fabulous presentation here. So everybody's muted so you don't hear all the clapping in the online. Thanks so much. So I assume there are a ton of questions, but in the moment everybody's muted, so you can unmute yourself. Jordan Smoller just unmuted himself, so maybe you start.
Thanks, Stephan. That was terrific and very exciting method. A couple of questions. One is, I think what you were saying was that the best situations are where you've got traits that are highly correlated and for which the mean chi-squares are pretty similar and also pretty large.
So they either need to be similar, or the chi-squared statistic for the trait you're interested in needs to be large. You can deal with one or the other.
So in your application, it's certainly, they're highly genetically correlated. It looked like the mean chi-squares are lower than the ideal threshold, but that just is that an indication of, well, first of all, am I seeing that right? And secondly, is that just an indication that it's hard to find some of those scenarios because our mean chi-squares are often pretty low?
Yeah, and so that's why I highlighted this. This is why it's an OR. So if we do the max FDR calculations in our specific setting, so the mean chi-squared for each of our traits is on the order of 1.4, and so, but it's equal across all of them, which means that there's a lot less potential for one trait to be pulling false information from the other traits. And so when you do the max FDR calculation in this specific setting with depression, subjective well-being, and neuroticism, you get numbers less than 1%. Recall that these max FDR calculations don't mean that this is actually the FDR. Like, the max FDR should be thought of like the worst case scenario. So if you wanted to find the genetic architecture that's going to screw me over, the very worst, the worst it can get is 1%. I suspect that it's actually less than that if we chose a realistic architecture.
So why is your recommendation as high as, greater than 1.7? That seems like a pretty stringent.
Yeah, so it turns out that if you have a mean chi-squared, so let's say that you're studying, like 7 different traits altogether, and let's say that the trait that you're interested in has a mean chi-squared statistic of 1.7. Then it actually doesn't matter if the other traits that you're studying have really large mean chi-squared statistics. You could be jointly analyzing with like height or something like that, which has a mean chi-squared statistic of I think 3 these days, or but you could even have something that has a mean chi-square statistic of like 10. And given that there's sufficient information in its own trait GWAS, these other traits are just not going to be able to pull the MTAG estimates away from the true value, even if they're really highly powered. And so 1.7 is not actually even that high these days in a post-UK Biobank world. I think that for moderately heritable traits in sample sizes, if it's available in the UK Biobank, you can get to 1.7 pretty easily.
Yeah, and can I just, while I'm here, ask one other question? Because as you said, the assumption of homogeneous omega is a pretty key one, and you very nicely described that, and we're very nicely kind of conservative about it. Can you just say another word again about the conceptual implications of what that means if the omega is homogeneous? Because it does seem like it certainly seems like it could be approximately true in many cases, but conceptually you could easily see how that might not be true, I think, right?
Yeah. So in my head, when I think about the homogeneous omega assumption, I'm primarily thinking about traits where the biological pathways are very similar. And so, so subjective well-being and depression, it might be that there are biological pathways that are distinct for those two traits, but it's kind of difficult for me to think of a story where that would be. But if there were cases where you could think of biologically distinct pathways, so I gave the example of cognitive performance and educational attainment. And educational attainment is a very complex trait that probably involves a lot of endophenotypes. So it might be IQ or grit or just anything that you can think of that might affect how much schooling a person gets, and a lot of those traits are going to be heritable. And so if you were to jointly analyze with MTAG education and IQ, you might run into problems because there are biological pathways that may be relevant for education that aren't relevant for IQ, and so that would violate the homogeneous omega assumption in that case.
But the very, very different individual betas for the different traits would not necessarily violate it. Is that true?
Yeah. So it is true that, like, in principle, you could have a homogeneous omega assumption, but still be genetically have a low genetic correlation, right? So a low genetic correlation could either mean that there are a lot of SNPs that are important for one but not for the other. It also could mean that all the SNPs that are important for one are the same for the other one, but just the betas vary widely. And if that's the case, then technically you're okay with MTAG. What I would worry about, though, is that you can't easily distinguish between those two. And so if you have a low genetic correlation, you're sort of in risky territory, because you could say, well, I think the homogeneous omega assumption's true, but then someone else could say, well, I don't. And then you're at a bit of an impasse. If you have high power, if your GWASs are high powered, you can often get away with a lower RG threshold. So I said 1.7 here as just kind of a rule of thumb. But if you had two GWASs that are both mean chi-squared 1.7 or something like that, then you could probably get away with having an RG much smaller than that, which is why I think which is why I recommend everyone doing their own max FDR calculations as well, because we can't search the whole space to make recommendations.
Great, thanks.
Are there others there? Unmuted?
Yeah, I have a question. Hey, Patrick, this is Claire.
Hi, Claire.
I was wondering, is there anything you can learn more specifically about where the genetic overlap is by comparing the change in the betas between the standard GWAS and then the MTAG GWAS? Does that tell you anything more than genetic correlation does?
Yeah, so that's an interesting question. So I mean, in principle, you could do that, and we've thought about it. I mean, MTAG's not really well designed for that sort of thing because it's built on this model, and so I mean, we're currently working on an extension to MTAG that will more directly answer that question, where we actually model these regions that are associated with one and not with the other. And so I feel like a model that's designed to pick out which regions of the genome are important for one trait or both traits would maybe be more successful at that. But yeah, I think that's true. And this is actually maybe another interesting thing that a person could do if you wanted to apply the method is if there's a SNP that is not associated with either trait, but then all of a sudden it's an MTAG hit, I think you can maybe trust that SNP than one that is not a hit in your primary trait GWAS, but is a hit in the secondary trait GWAS. Because if it's a really strong association in your secondary trait, and then all of a sudden you find that it's like marginally significant in the MTAG results, you might worry that this is actually just one of these bad SNPs where it's important for trait one but not or important for the secondary trait but not associated with the first one. And so there are some things that you could do, I think, kind of post MTAG analyses to explore the credibility of your findings. We've not looked into that carefully though.
Okay, thanks very much.
Somebody else who has questions here? We still have 40 people on the call.
Hello, this is Eric Johnson. Can you hear me?
Yes,
yeah.
we can.
Okay, Patrick, a couple of questions. One, following up on what you just said. So if the way I understood it, so if the primary GWAS has a stronger signal than that MTAG GWAS, what was your interpretation of that?
Well, I mean, so I was just saying, so let's say, so neuroticism, for example, has a region on chromosome 7, I think, I forget, but it's associated with this inversion, and it's this enormous peak. It's like way an outlier relative to everything else in that GWAS. We removed it from MTAG because it was such an outlier that we thought that it would drive these results. But so what you would probably find if you jointly, in fact, what you do find, we did this, if you don't remove that region and then apply MTAG, is that all of a sudden that inversion region is strongly associated with all of the traits. Now I find myself really skeptical of those results because I know that that's a region of long-range LD with abnormally large effect sizes for neuroticism, and so it makes me think that even though there's an MTAG depression hit in that region, I don't really trust it as much because it didn't exist in the depression GWAS and it was enormous in the neuroticism one, and so I would think that particular SNP is maybe less credible than one where you couldn't see it in either of the traits and all of a sudden you see it in the MTAG results. Because what that means is that it probably was almost significant in both of them and with a consistent direction and sign and that kind of thing. And so once you look at them together, MTAG says, oh, well, since we have two things that are almost significant in a pair of traits that are highly correlated genetically, that's pretty good evidence that it's actually associated with each of them.
Right. I guess I was thinking of it in somewhat the other direction where you have a significant hit in your non-MTAG GWAS, and then you apply MTAG when you think you have correlated phenotypes, and that signal goes away. in the MTAG results.
Oh yeah, yeah. So that can sometimes happen, and I think that does happen. We do lose a couple. You know, it's hard for me to think of exactly how you might interpret that. I mean, it might just be like a power issue, right? Like any SNP that you find, especially like when it's significant in the GWAS, then there's, you know, the true effect size might, in fact, will on average be less than what you estimate it to be. And so when you jointly analyze it with something else, it might disappear. I mean, you see the same kinds of things happening if you do meta-analysis, right? So if you're trying to boost sample size, some SNPs may disappear, and that doesn't mean that they weren't real in the first place. It's just that, you know, these are statistics and they kind of move around due to sampling variance.
Right. And if I may, one other question. You talked about or mentioned that the approach is robust to sample overlap. How robust? I mean, if you take an extreme example and say you were to look at multiple phenotypes with the same cohort, so the overlap is almost complete.
Yeah, it should still be fine, even in that case. We did some simulations in the paper. Do I have, let me see if no, I didn't include those slides here, where we kind of made synthetic overlap in the UK Biobank. And so, you know, we did GWASs of the same trait, but we made sure that there was a large amount of overlap between the summary statistics. And the z-scores that MTAG produced were like nearly perfectly correlated with the z-scores that you got if you just did a GWAS of the entire UK Biobank, you know, not doing MTAG on these overlapping subsets. Okay. So it seems to work pretty well.
Thank you.
Thanks so much. I think so. We are 3 minutes before the hour. So if they're not like some very pressing questions, somebody here who wants to ask. I mean, Patrick is always online, so we can ask him via email as well.
Yeah, feel free to reach out to me if you have any questions. I'm happy to chat.
Exactly, that's exactly what I meant. So to summarize this, I think so. I mean, it's a fabulous new method that's coming out here. It's highly thrilling, but obviously, and that's exactly what Patrick also emphasized today. It's not like just put everything that you find as summary statistics together, run MTAG and publish it as it is, right? So there are some scenarios that you really want to have a close look, and I think we will probably need Patrick and his colleagues for a while to actually find out exactly the right scenarios where we run these things, and we stay a little bit cautious about what we're doing, obviously, as always, but it's great to have that, and it was a fabulous presentation.
Thank you.
And if I count now down to the full hour, but anybody else? Last chance for a question? No, but great. So we got to the full hour. I wish everybody a great morning, afternoon, evening, whatever it is. Have a good one. Thank you very much.